Welcome to chapters 10 and 11. This first chapter is going to be talking about muscle physiology and the way it actually functions and how it integrates with the nervous system. And then chapter 11 is basically an atlas of muscles, and I'm going to be pointing out to you the ones that you need to know for testing purposes. This is often people's most difficult chapter. I get my worst test grades on this chapter. It's very important that you watch the little video clips that I have put on Blackboard for you to try to help visualize how this muscle physiology takes place. As a review, you saw this before in chapter 4. You have two types of muscles, striated and non-striated. Striation refers to a stripe. So all they're saying here is that under a microscope, cardiac and skeletal tissue appear to have stripes. Smooth muscle does not. So make sure you know that of the three types of muscles, the striated versions are skeletal and cardiac. We'll be doing the bulk of our discussion about cardiac muscle when we get to chapter 20. It is involuntary. So as opposed to skeletal being striated and voluntary, cardiac is striated and involuntary. And then in later chapters also, we'll talk more about smooth muscle. It is also involuntary. You'll find smooth muscle lining all the blood vessels. You'll find smooth muscle lining the hollow organs, such as the lungs, the gastrointestinal tract, the reproductive tract. So the bulk of this chapter is going to be talking about skeletal muscle. So the skeletal muscle system, again, is striated and voluntary. It uses the bones as a lever to create movement. Muscles always cross joints. You're not going to find typically a muscle that originates and inserts on the same bone. For example, instead of having a muscle that originates on the humerus and inserts on the humerus, you might find one that originates on the humerus and inserts on the radius or the ulna. So they cross joint so that when they shorten, they contract, they move that joint, and now they've created movement. Remember, please, that they are controlled by the somatic nervous system. So when you see the word somatic, you need to think skeletal muscle. You need to think voluntary. So again, here's simply a diagram of cardiac muscle tissue. It looks striated, so if you look closely on the slide, you'll see tiny little, if I can stabilize this slide for just a moment. No, it's not going to. Let me try it this way. You'll see the tiny little stripes in here. You actually have a specialized cell junction type called intercalated disc, and these are what you see with the darker stripes. So the intercalated disc allow the information being passed through the nervous system for the cardiac function, basically, to be passed very quickly from cell to cell. It also provides a security, and as far as when the heart muscle contracts, it's not going to allow the cells to be pulled apart or disrupted. And again, please remember that the cardiac muscle tissue is involuntary. And you can notice here on this photomicrograph of smooth muscle, you see none of the stripes that you saw earlier. It is involuntary, and it again lines your blood vessels. It'll allow vasoconstriction, vasodilation, in other words, opening and closing of the blood vessels to maintain blood pressure. And it lines hollow organs. Think about the fact, we haven't talked about this yet, but we will in 142. When you eat something, it's going to have to be pushed or propelled all the way down from your esophagus through your stomach, through your intestinal tract. And that is a function of the smooth muscle located there. But again, for the bulk of this chapter, we're going to be talking about the skeletal muscle system. And in general, when you hear someone mention the muscular system without specifying, they're going to be talking about the skeletal muscle. Remember that it's going to be tendons that attach your muscle to the bone. So ligaments attach bone to bone, tendons attach muscle to bone. And by the way, these tendons happen to be extremely tough. Every now and then, if you have somebody with traumatic injury, instead of actually tearing or rupturing the tendon, it will actually break a section of bone loose. You've seen some of this before. Again, your functions of skeletal muscle producing movement. That includes the function of breathing. you have the muscles between your ribs, your intercostal muscles, and in particular, the diaphragm, that when they contract, it expands the thoracic cavity, which is going to allow air to rush into your lungs. We'll talk about that a whole lot more in chapter 23, but that is a reflexively controlled skeletal muscle function. You may not think about the fact that your postural muscles are always keeping you upright. They are voluntary, although you may not think about it because you're not sitting there concentrating saying, gee, I need to hold my head up, or I need to hold my spine upwardly aligned so I can sit in a chair. But if you don't think they are voluntary, see what happens, for example, if you've ever been on an airplane and you doze off and you're sitting there doing the bobblehead and you lean on the guy next to you and you drill on his coat. Those are what happens when you lose the voluntary control of those posture muscles. Your abdominal muscle is actually about the only ventral protection of your abdominal organs. So that's why, for example, in some sort of car accident, a lot of times you'll see things like ruptured spleens, liver lacerations, because instead of having a bony cage like you do for the thoracic organs, you simply have the abdominal musculature, but it's better than nothing. You're going to have skeletal muscle that is going to be around the openings of your GI tract and also around the urethra. So, for example, you have your orbicularis oris, which is a skeletal muscle around your mouth that allows your mouth to open and close. You also have an orbicularis anus, which is basically allowing you to control when you choose to have a bowel movement. And then you also have the same thing for the urethral sphincter. So you actually have, for those last two examples, you have two sphincters, an internal sphincter and an external sphincter. The internal sphincter is always smooth muscle and it's involuntary, but then you have the external sphincter, which is going to be skeletal, and it's always voluntary with any luck at all. Body temperature is maintained by skeletal muscle. That's why when you get really cold, you'll start to shiver. You have these tiny fasciculations, these tiny tremors of your muscles. What's happening when you shiver is you're contracting your agonist and antagonistic muscle, we'll define those terms later, at a very high oscillatory rate, a very high rate of very tiny contractions. And what that's doing, that's breaking down ATP, it's releasing energy, and 60% of the energy is given off as heat. So, in fact, think about when you exercise. If you're doing a lot of physical activity, you get hot and you'll start to sweat. If you're sitting doing nothing on a chilly morning, you might get uncomfortably chilly, you might get cold, and you have to get up and walk around. When you walk around and move around, your muscular movement has increased heat production, and therefore you feel more comfortable. So skeletal muscle also provides a storage of things like glycogen. We've talked about glycogen a little bit. That is the storage form for carbohydrates in animal tissue. Also amino acids. We've talked about the fact that amino acids form proteins. So if your body is in a starvation mode, if your body has used all its carbs, it's used all its fats, then and only then is it going to choose to break down skeletal tissue. Skeletal tissue is difficult to break down for your body because it's going to require a lot of oxygen. It's also going to release a lot of nitrogen, which the body is going to be having to get rid of in the form of ammonia. So too much nitrogen in your bloodstream, it can put you into a coma and can kill you if it's not properly maintained. So it's certainly not your body's first choice of nutrient sources to metabolize. You may have seen photographs of starving children in famine situations such as Somalia, Sudan, and what you may have noticed is that beyond the fact that their arms and legs are extremely skinny and stick-like, but their abdominal area will be very round and protuberant like a little pot belly. And people mistakenly think, oh, that means that they have food in their abdomen. No, what it means is that their body has metabolized or catabolized their skeletal muscle, and so they've lost that layer of abdominal muscles that would normally keep your intestinal tract kind of pushed back into place. So it's simply that there's not a whole lot between the outside world and their internal viscera. So that's why you see that little round stomach on starving children. Let's cover some basic terminology before we get too far into this. A muscle cell is the same thing as a muscle fiber. Those words get used interchangeably, so don't be confused by that. Muscle cells are multinucleate, meaning they have many, many nuclei. And the reason for this is if you notice this word, myoblast, anytime you see the suffix blast, that means an immature form. So a myoblast is an immature muscle cell. The way muscle cells form is that first off you have what they call a myosatellite cell that turns into a myoblast, and then the myoblast fuse together to create this larger muscle fiber. You have some muscle fibers in your body that will be very, very long. So think about the fact of a long muscle fiber, for example, in the calf of your leg, or in your quadriceps femoris in your thigh. So some of these muscle fibers are pretty large. The reason that you have all these nuclei is your nuclei are the location of your DNA molecules. So your DNA, remember, is a blueprint for protein. And muscle is primarily protein. So as you grow and as you build muscle from any sort of exercise, that sort of thing, that muscle actually grows relatively quickly. And because you've got all that DNA, you can reproduce that protein molecule many, many times faster than what you could if you only had one nucleus. The example I always give, think about somebody building a skyscraper. If you've got a 90-story building to build, you're not going to have one set of blueprints located on the ground floor. You're going to have a set of blueprints in multiple locations on each floor, so that the construction workers have something to refer to very quickly so they can build the building much more quicker. And again, that blueprint would be your DNA. Make sure to know the term fascicle. A fascicle is simply a bundle of muscle fibers or muscle cells. So fascicles themselves then bundle together to form the entire muscle body. So if you look at this, this is representing a muscle cell. You put a bundle of them together and you've got a fascicle. Then you put fascicles together with other fascicles and you've created an entire muscle. So for example, what they're representing here would be the extensor carpi radialis. So it's just simply a bundle of fascicles. Now on the previous slide we went upwards as far as size. Now we're going to go downward a little bit. Each individual muscle cell itself is wrapped by endomysium. So the endomysium is going to be the location of the myosatellite cells I referred to earlier. So the endomysium is going to be the smallest layer of connective tissue covering here. You're covering an entire muscle cell here. So then you put the muscle fibers together, you've got a fascicle, and the fascicle is surrounded by the perimysium. When you put the entire muscle body together, you've lumped all those fascicles together and now made it an entire muscle. That's going to be surrounded by a connective tissue layer called epimysium. So endomysium, perimysium, and epimysium. So make sure you relate, for example, endomysium is a connective tissue around the muscle fiber or muscle cell. Perimysium is around the fascicle, and epimysium is around the entire muscle body. So what you're going to have out here is going to be fascia, blood vessels, nerves, and then other muscle bodies around it, depending on your location. So again, those are three definitions you do need to know for testing purposes. Some more terms that you need to be familiar with. Number one is going to be sarcolemma. The sarcolemma is going to be the surrounding of the cell or the cell membrane or plasma membrane. Muscle fibers are made of bundles of what we'll talk about in a few minutes, myofibrils, put together. So you have these bundles of these fibrils that are lumped together. So it's quite a complicated internal arrangement inside the cell. The cell needs a way to communicate with all the internal bundles. It does that by having these invaginations of the sarcolemma into the cell, and that's called a T-tubule. So the T-tubules are actually extensions of the sarcolemma deeper into the cell. And we'll be talking about why that's important in just a few moments. The sarcoplasmic reticulum is the same thing as the endoplasmic reticulum, and it plays a very important role in muscle physiology. It's going to be storing calcium ions. Very important concept there. So terminal cisterna is going to be a dilated end of the sarcoplasmic reticulum. And so when we get a closer look in just a moment, what you're going to see is they'll talk about a triad. They'll be talking about this enlarged end of the sarcoplasmic reticulum, called the terminal cisterna. And then the other part of that is going to be the transverse tubule. And then on the other side of that, you've got the other terminal cisterna. So those are the three things that make up the triad there. The last definition on here is going to be sarcoplasm. The sarcoplasm is the same thing as the cytoplasm of the cell. So maybe you've picked up by now that the prefix sarco means flesh or muscle. The term for a cancer of the muscle, which is actually fortunately fairly rare, but it's called a sarcoma. So taking a closer look, this is simply a small portion of a cell. This is representing your sarcolemma there, your plasma membrane. Here are the T-tubules that are the extensions of the sarcolemma going deep into the cell. So your sarcoplasmic reticulum is represented here in purple. The terminal cisterna is this dilated end. And then here are all the little myofibrils that we'll be talking about in just a moment, bundled together. The problem that we have in talking about the muscular system is that to do so, we have to bring in the nervous system. And we haven't talked about it yet, and so I think that might be one of the reasons that people have difficulty on exams trying to totally be comfortable and understand the whole principle here. So let's go ahead and look at the neuromuscular junction, basically where the nervous system meets the muscular system. In a nutshell, it's going to be your nervous system that's driving the muscular system. So it's going to be having to transmit information from the brain or the spinal cord down the nerves into the muscle to tell it what to do. So this is representing the end of an axon. And again, it's difficult because we really haven't talked about nerves yet. But if you recall, a nerve cell or neuron will look something like this. And you've got dendrites that carry information inward to the cell. And then you've got axons that carry information down the cell. At the end of the axons are the little branches called teledendria. And don't worry about this right now. We'll get to all this when we get to chapter 12. But at the very end of this, you have what they call synaptic terminals. So this is the part that we have to worry about right now to be able to understand what we're looking at. So the axon is this part right here. The synaptic terminal is this little bulb, and it contains synaptic vesicles. Synaptic vesicles contain a chemical referred to as a neurotransmitter because it's transmitting information. An area where two cells meet to communicate is referred to as a synapse. And we'll talk about synapses again a whole lot more in chapter 12. A nerve cell can meet a muscle cell, or it might meet a gland cell, or it might meet another nerve cell. Where they meet is called a synapse. For our purposes right now, we're talking neuromuscular synapses. So this entire area is going to be your synapse. Go ahead and notice that there is a space between the muscle cell, which is this part, and the neuron, which is this part. That is referred to as a synaptic cleft. So they don't actually meet. The motor end plate, make sure you pick up this because I guarantee you a test question on this. The motor end plate is a part of the muscle that faces this synapse, and it's where you're going to have the receptors for the neurotransmitter. So, and just to be finishing up our list here, the terminal cisterna, that's the enlarged areas right here, part of the sarcoplasmic reticulum, and then the T-tubule is this part right here. I sometimes have this picture on an exam to label, so make sure you can do that if I ask you to. So what's happening here, you have information being transmitted from the neuron down the axon. It's going to be causing these vesicles containing neurotransmitters to dock on the end of the membrane here. It's going to release the neurotransmitter into the synaptic cleft. The neurotransmitter is going to bind to receptors on this motor end plate, and that's going to cause the information to be passed down the sarcolemma and down the T-tubules to tell the muscle to contract. Now here again, we're going to have to talk some neurology before we can continue to talk about muscle function here. An action potential is basically an electric current that's transmitting information. We'll be talking about action potentials a whole lot more in chapter 12, but we have to be able to define it right here. So an action potential is basically an electric current. Remember that the nervous system uses electricity to transmit information. And the way it's able to do that is you have what they call a resting membrane potential, which means that you've got slightly more negative charges on the inside of the cell than what you have on the outside. And so what happens when this thing depolarizes, you're having openings of little channels that are letting sodium ions come in, and then you're going to have openings of other channels that are allowing potassium ions to go out. So what's happening as you go down the cell membrane, you're actually reversing the charges. You're going to be making the inside of the cell slightly more positive than it should be, and the outside of the cell slightly more negative. When you do that, you were said to have depolarized that membrane. So after you depolarize, you've got to repolarize, and then there's going to be a very short phase called hyperpolarization that we'll talk about again a lot more later. But if you look at this graph, what's happening here is there's a threshold of charge that you have to cross before you hit an action potential. An action potential is an all or nothing proposition. There's no such thing as a small or large action potential. What's happening here, your resting membrane potential is at this level. So the cell is sitting here doing absolutely nothing, totally content at minus 70 millivolts. Okay, something happens here and causes a stimulus, and that stimulus is going to start reversing the charges that we talked about up here, and it's going to depolarize that cell membrane. Now, if it only comes up to here and comes back down, it doesn't hit the threshold, it doesn't fire, it does not create an entire action potential. And again, we'll cover this a lot more detail later, but I have to go through the basics to talk about muscle here. So, however, if the stimulus is big enough that it causes a depolarization that goes past the threshold, now you produce an action potential. So this is all going to be sodium channels opening up and sodium ions fluxing into the cell. This point, you've got sodium channels being inactivated. Now you've got your potassium that's actually leaving the cell. And down here, you've got everything. This is basically a potassium function down here. We'll talk about this a lot more later. Right here, you become more negative than you were at your resting membrane potential. So at the end of hyperpolarization, you've returned to your normal resting membrane potential. So that in a nutshell is an action potential. Again, you can't have a large or a small one. I always say it's like being pregnant. You either are or you aren't. You either have an action potential or you don't. So that action potential is going to be transmitted from a nerve into a muscle cell to cause contraction. So here's a better graph showing what an action potential looks like, and it's got it broken down. This is a chart that you're going to need to know, at least for the nervous system part. But again, what this is showing here is that you've got a stimulus being applied. This is representing your resting membrane potential. Stimulus is applied. You've got your depolarization. You've crossed the threshold now, so you've peaked out this action potential. You've got your repolarization, your hyperpolarization, and then you return to resting membrane potential. So this happens in both nerve cells, and then it happens also in muscle cells. And this is how information is being passed from the nervous system to the muscle cell. After you've had the action potential, you're going to have to return those ions to the proper side of the membrane to restore that resting membrane potential. So what's going to happen is you're going to have a molecule that is located on the cell membrane that's referred to as the sodium-potassium exchange pump. Your book may refer to it as a sodium-potassium ATPase exchange pump or however, but I just say sodium-potassium exchange pump. So it's going to be this pump that's going to be pumping the sodium back out of the cell at the same time that's pumping the potassium back into the cell. It's not totally an even swap, so you've got 3 sodiums going out, you've got 2 potassiums coming in. But it's going to be that pump that restores that resting membrane potential, and this pump requires ATP to function. So adenosine triphosphate, if you recall this molecule, is a nucleic acid that stores energy. So for this to be produced, you have to have oxygen. If you stop breathing, you can't produce ATP. You have no energy. You cannot restore the resting membrane potential, and nerve tissue and muscle tissue ceases to function. This was just a diagram that I found showing that pump in action. So here it is. You're swapping the sodium for the potassium. Again, here's your cell membrane, and here is your pump. Okay, let's go a little bit deeper in naming the muscle cell parts here, switching gears a bit. This represents a short piece of a muscle cell. Inside that muscle cell, you're going to have a structure referred to as a myofibril. So if you look at this wrapping that you're seeing right here, that is your sarcolemma. So that's your plasma membrane. Here's a cell nucleus. So there's another nucleus that you see. There's another, remember the skeletal muscle is multinucleate. And so what you've done, you've taken a long muscle fiber and you've cut a section of it, and that's what you're looking at right here. So the myofibril is made up of these tiny little things called a myofilament. So going back to organizing levels of organization here, make sure you understand that myofilaments form a myofibril. Myofibrils are forming the muscle fiber. So the myofilaments we're going to be talking about, you need to know these, are going to be actin and myosin. So also make sure you know that actin is referred to as the thin filament, and then myosin is referred to as the thick filament. So they are arranged in these little short units called a sarcomere. So the sarcomere is just this tiny segment of a myofibril. So just to make sure you understand this, myofilaments, such as actin and myosin, are these tiny little bands in here, and they create the myofibril. You put a whole bunch of myofibrils together, you have a cross-section here of the muscle cell. Remember, when you put a muscle cell, also known as a myocyte, together, then you've got a fascicle, you've got other muscle cells in there with it, and then you put the fascicles together and you've got the muscle body. So this is what we talked about earlier in the chapter. So go back to that if you need it. The only thing we've added to it is the myofilaments and the myofibrils. Now, please don't try to memorize these numbers because I'm not going to ever ask you this. I put this in here so you've got some sort of comparison in your brain here. So a single muscle cell can have up to 15 billion myosin filaments. Each myofibril, again, the myofibril are represented by the little dots that you see over here, contains about 10,000 sarcomeres. So what we're going to be looking at is going to be on a very, very tiny scale. It's important that you know that a sarcomere is the functional unit of muscle tissue. The sarcomere is a functional unit of muscle tissue. It's described with bands and zones and lines based on the way it looks under a microscope. If you were to lace your fingers together, in other words, really my art's bad enough, but really when you put the stylus factor in here, but interlace your fingers like this, ooh, that is bad, and then slide them back and forth. So what you're representing here, this area, would be a sarcomere. So now if you slide your fingers back and forth, so if you're actually, again, moving your hands toward each other, that would be similar to the way a sarcomere contracts. So your fingers, when you move them together, so they're totally interlocked with each other, your fingers aren't getting any shorter, but the area representing the sarcomere is. The same token, these lines that you see representing the actin and myosin filaments do not get shorter. They do not contract. They simply slide across each other so the sarcomere gets shorter. Now, I typically never have any test questions about the bands and the zones and that kind of thing because I really don't think it's that important. The only thing I might ask you to say is the Z bands being on each end of the sarcomere. So, but again, they talk about isotropic, anisotropic, and that's what they talk about, I bands and A bands, the H zones. But again, what it's talking about, if you picture the area of overlap of your fingers when they're contracted, when they're put together or not put together, that's what creates the appearance of these zones. So this M line is representing the center of a sarcomere. This Z band right here is representing the bracket. And so a sarcomere is going to be located right here. So this darker area is going to be representing the myosin. And then where it's not with a lighter area is simply actin. So again, for testing purposes, that's not that important. But I really want you to be able to picture these things being lined up and contracting together and then sliding back apart when the muscle relaxes. Make sure you know and make sure you understand the concept that it is a sarcomere that contracts or shortens in length. The filaments themselves, the actin and the myosin, do not shorten. So here you see it on a little bit cleaner diagram as opposed to a photomicrograph. Again, here's your Z lines. This entire area is representing your sarcomere. And what's happening they're sliding together here in this bottom one, and they're contracting. The muscle's getting shorter. When a muscle contracts, it produces movement. So we need to talk a little bit about how do the fibers, and how do the filaments, I should say, know when to slide across each other. The sarcoplasmic reticulum, remember this is the same thing as the endoplasmic reticulum, stores calcium ions in the enlarged area referred to as the terminal cisterna. When the action potential travels down the axon, and you've got transmission of that action potential down into the cell. There's your T-tubules. Let's draw some little terminal cisterna and endoplasmic sarcoplasmic reticulum. So the red is representing your sarcolemma. The black is representing your sarcoplasmic reticulum. And the blue is representing the axon. You see now why I don't make my living as a commercial artist. So, again, you've got calcium that is stored inside there. So let's draw one more thing in here. Let's draw some filaments. It would actually be under the sarcoplasmic reticulum, but we're going to be easy here and draw it like that. So what happens now, you've got the action potential, that reversal of electric charge coming down the axon from the neuron, down into the synaptic terminal. You've got the neurotransmitter coming out of the vesicles. You've got the neurotransmitter binding to the receptors on the motor end plate. And now you've got an action potential being passed down that sarcolemma, down the T-tubules, into the muscle cell themselves. And what's going to happen now is you're going to have this sarcolemma contract and shorten. And we'll talk about how the calcium does that in just a moment. But understand the action potential is coming down the axon and being carried through the sarcolemma, down through the T-tubules, and is causing contraction of the sarcolemma. So let's look closer at these myofilaments here. I have used, by the way, both of these diagrams on this page on exams, so you do need to know how to label. I'm not worried. I really don't care about troponin T, C, or I, just know troponin. Don't worry about alpha tropomyosin, that sort of thing. But what's happening here, this is representing the myosin. This is representing the actin. So what's happening here is going to be an interplay of the calcium on the troponin. So here is your representation of your sarcomere. Again, what's going to happen is you're going to have a contraction of the sarcomere when these filaments start sliding across each other. Now, I've got it broken out into multiple slides. We're going to talk about it several times over, but just to give you a preview, what's happening here, the calcium that's been released from the sarcoplasmic reticulum is going to bind to the troponin molecule. The troponin molecule is going to cause this tropomyosin to rotate off. It's going to open up a binding site that's going to bind to this myosin head. And then this myosin head is going to actually pivot and pull the actin down. Now, I've got some great little videos that actually show you this in a stepwise sequence. This is almost impossible to talk about without having some sort of animation. So please, after we start talking about it a little bit more, go ahead and take a break and look at your animations, because it's really important you're able to visualize this. So again, here's your actin, here's your myosin. Make sure you know that it's the myosin that has these myosin heads that are going to be binding and forming what they call a cross bridge. So here are the steps of contraction, and this is referred to as a sliding filament theory of muscle contraction. You need to understand these steps enough to be able to place them in order on the exam to put them in proper sequence for me. So if you visualize this a step at a time and watch the animations, then you should be able to understand this. So number one, you've got the arrival of an action potential. It's going to be passed down the sarcolemma and distributed through the cell by the T-tubules. You're going to have the release of the calcium ions from the terminal cisterna of the sarcoplasmic reticulum. These calcium ions are going to bind to the troponin molecules that are located on the tropomyosin that's wrapping around the actin. That's going to pull the tropomyosin filament away, and it's going to expose binding sites. So again, sort of picture your actin, and then if we were to draw the tropomyosin kind of wrapped around it, put your troponin molecules on here. Now just picture again what's going to happen. The calcium is going to bind to the troponin, and it's going to pull this whole tropomyosin kind of away. It's going to kind of rotate it in that direction. And that's going to expose the binding sites. And then what's going to happen, the myosin heads are going to come up and bind to the binding sites. So draw the myosin down there. So now we've got binding of those myosin heads, and you have now formed a cross bridge. You have a link between the actin and the myosin. Then you have a ratcheting of the heads, in other words, a flex, and that's called the power stroke, and it pulls the filaments closer together, and it shortens the sarcomere. There is a DVD that came with the Martini book. It is on reserve in the library, and it has a decent little step-by-step animation of this. It kind of breaks it down and it goes into a little bit more detail than what I care about, but again, it's a step-by-step animation that might help you be able to visualize this if you're having trouble with this. So now you've got the power stroke. So you've got these filaments pulled closer together. You've got the sarcomere that's going to be shortened. You have muscle contraction. So now the next step is to break down the cross bridges, and that's broken down by the formation of ATP. So this is where the energy release and the heat production happens in muscle contraction. The myosin heads get basically reloaded or returned to what they call the high energy position. Calcium ions go back into the sarcoplasmic reticulum, and the muscle has now relaxed. So those 8 steps you need to be able to put in order, talking about the sliding filament theory of muscle contraction. Understand, especially this bottom point right here. Contraction is an active process. Relaxation is passive. In other words, when you're moving a muscle, for you to actually do anything, you contract it. It relaxes when you stop contracting. So this takes energy, it takes ATP, this does not. Contractions only happen for as long as you keep sending action potentials down that nerve. So eventually, you're going to run out of ATP, and you're eventually going to run out of calcium, and that muscle fiber can no longer contract. I put this photograph in there to remind myself to tell you about rigor mortis. I'm sure you heard the term rigor mortis before. It's the stage, a temporary stage, of stiffening of muscles after death. This happens because, I referred to this earlier, when you're not breathing, you cannot generate adenosine triphosphate or ATP, and when you can't make ATP, you don't have ATP to bind to those cross bridges to break them down. So after death, within a matter of a few hours, the cross bridges in your muscle cells can't be broken down, so you have this stiffening. This stiffening is going to last until the enzymes in your cells get broken down from the peroxisomes and all the lysosomes in your cells, and you basically start to undergo the process of what they term autolysis or self-digestion, and that breaks down the membranes and the body softens back up. So you really, there are experts that can tell you exactly how long rigor mortis is going to last, but it's going to vary based on ambient temperature. In other words, what the temperature is in the environment, the body size of the person, that sort of thing. There are a lot of variables that go into that. So this is a point, if you want to take a break, this would be a good stopping point. We're going to start talking about reflexes a little bit after this, and I'll see you soon. I want to go ahead and mention two skeletal muscle reflexes here. We'll talk more about reflexes in the nervous system. Now, I noticed in the online version of the free text that I put the link to, I really couldn't find a whole lot about muscle reflexes in particular, so that may be something, if you're using the free online text, you may have to find an outside source. There's plenty of material, everything from Wikipedia on down, but I just wanted to describe two muscle reflexes here. One involves what they call the Golgi tendon organ. The Golgi tendon organ, just like it sounds, is located inside the tendon, and it's designed to prevent the muscle and the tendon from overstretching. So basically, think about trying to pick up the bumper of a car, while it's still attached to the car, and you're expending this huge amount of force on a load that you really can't pick up. It's going to stretch that tendon, and it's going to cause a reflexive loosening of that muscle tension. So it's inhibitory. It's going to actually inhibit the muscles in an effort to keep you from overstretching the muscles and tendons. The opposite of that is going to be caused by a tiny organ inside the muscle itself called a muscle spindle. And this will actually, when it's activated, cause a contraction. So you'll see this referred to sometimes as a myotactic reflex. So if you've ever seen the example of the patella tap reflex, the physician or whomever takes the little reflex hammer and taps on the tendons below the patella, and that causes a rapid stretching of that tendon, and that causes a reflexive contraction of that muscle, which means that your foot kicks upward because you're shortening that muscle. So that's muscle spindle reflex is a myotactic reflex, and it causes the muscle to shorten. The reflex that I spoke about with the Golgi tendon reflex, that causes it to actually lengthen. Sometimes you'll see that referred to as the inverse myotactic reflex. This diagram is representing basically a bundle of muscle fibers. These are called extrafusal on each side because this is called intrafusal. It's simply a specialized bundle that contains the stretch receptors. So we'll interplay this with the spinal cord when we get to chapter, let me see, I think it's 13. What we'll be looking at basically a whole section on spinal reflexes, but what's happening here, this is showing again your Golgi tendon organs located inside the tendon. You've got some kind of massive stretch going on here. It causes an arc to go through the spinal cord, which feeds back to the muscle body itself, which is going to relax the tendon. So right here, you've got some sort of massive pull, and here you've got a counteracting relaxation, and that is the Golgi tendon reflex. Okay, I've added a slide here because I wanted to give you a definition that I realized I did not have in the slides, and that's tension. Tension is defined as the force that is generated by shortening the sarcomere. And this is done in response to a load. When a muscle contracts, it's doing so in order to perform work to move a load. So tension is produced by the muscle attempting to move the load. The next couple of slides I've actually moved earlier in this presentation If you've got the printouts from the PowerPoint, you'll actually find these next two slides further down, so bear with me on that, but I'm trying, every time I use these slides, I end up moving them around, trying to make them a little bit more cohesive. There are two types of skeletal muscle tension that I want you to know these definitions for: isotonic and isometric. Isotonic is when you're picking up something that you can actually pick up. The force is greater than the load. Picture the load being a gallon of milk. It's something that, let's say it weighs 2 pounds, and you're able to easily pick up that 2-pound gallon of milk. Now, let's look at isometric. Isometric is when the load is greater than the force. Let's say you're trying to pick up your car. So the load, the weight of that car, is greater than the amount of force that you can create with your muscles. So the way to actually break these words down, iso means same. So tone means the same force, the same amount of tension to overcome a load. If you pick up that gallon of milk and you hold it at the level of your thigh, it's going to be 2 pounds of a load. Let's say you flex your elbow and now you're holding that gallon of milk even with your waistline, and it's still 2 pounds of load. Now let's say you extend your arm up over your head. You've got that milk gallon up above your head. It's still 2 pounds worth of load. So the load does not change. It's going to be the length of the muscle that changes. So to fully understand that, you need to be equating tonic with tone, and that being basically the amount that's overcoming the load. Now, isometric, iso, same, metric meaning length. So picture you're pushing on the wall. Well, you probably can't move it. If you do, you need a new wall. But let's say you're pushing on your wall lightly. You're only generating about, say, 5 pounds worth of force. But the wall's not moving. You can't overcome that load. So the length of your muscle is not changing. Now, push on that wall as hard as you can. Let's say you're generating 30 pounds worth of force. Wall's still not moving. The muscle hasn't changed length because the wall has not moved. So the length of the muscle is staying the same. Now, you may see in your book when they're talking about isotonic contractions, they'll be talking about concentric contractions and eccentric contractions. All they're talking about there is concentric. Let me clean that up a little bit so that's readable. Okay, with a concentric contraction is when you're going upward and you're shortening that muscle. And with the eccentric is when you're coming downward and you're actually lengthening that muscle. So this slide gives you a good example of what I'm talking about. This entire page is only talking about isometric contraction. There's nothing on here about isotonic, okay? So your concentric is again shortening, and then your eccentric is going to be lengthening that muscle. So the tension that's going to be produced in a muscle fiber is going to be dependent on a couple of different things here. Number one is going to be the starting length. There is actually what they refer to as an optimal starting length. It's said to be between 75 to 130%. Now some books will say 80 to 120%, but that's pretty close. So what they're saying is if you start out with sarcomeres that are already pretty extended, you've got the muscle fairly lengthened, then there's not very much overlap to allow this to really grab, to allow a high enough number of these myosin heads to interact with the actin. So there's not a whole lot to grab a hold of to pull these closer. On the other end of the spectrum, if you've got sarcomeres that are already contracted, you've got a muscle that's almost as short as it's going to go, there's also not a whole lot of room here for movement. So the ideal is going to be right here in the middle. So if you've ever taken weightlifting class. Before I started teaching, when I was healthier, I was going to the gym 4 and 5 days a week, and I always enjoyed what they called back then, it was just a proprietary name, a group power class. It's where you do a series of exercises for one muscle group during the length of a song. So you might be exercising your triceps for 5 minutes or your biceps for 4 minutes or whatever. To give you the example of what I'm talking about, though, as far as the optimal length, when you first start exercising your biceps, you start with your hands almost down by your thighs. Your arms are almost fully extended. You've got a barbell or a weight bar in your hand. And then what they'll do is they'll tell you to contract. So you lift that bar upward and bring it up to your chest and a biceps curl. Then you repeat that for a number of times. Then to vary the exercise, they'll tell you, okay, keep it in the middle. So what happens then, you'll drop your barbell down to about half that normal extended length. So you've actually got your forearms parallel to the ground. So your elbows are held at a 90 degree angle. And so then when you're doing your biceps curls from that 90 degree angle, it's a lot more fatiguing. And the reason for that is you're not allowing a whole optimum length of that sarcomere to give you enough tension to make it easier to move that load. So I just want to kind of throw that example at you. But there is an ideal mid-range of sarcomere contraction, that's your starting point, to give you the ability to be able to produce a maximum amount of force. Now in this next slide coming up, we're going to be looking at other things that can affect the tension, and that's going to be having to do with how often you actually stimulate the fiber to contract. So the stimulation of that muscle fiber to contract is caused by the frequency of stimulation. So we talk about a muscle twitch. Understand that I'm not talking about a twitch that you might get, for example, in your eyelid or in a finger. Those twitches that you're actually able to see are actually the summation of lots and lots of tiny muscle fiber twitches. The muscle fiber twitch that we're talking about is an individual muscle fiber, and again, just by itself, you'll be totally unaware that it's even happening here. So if you look at this twitch is only lasting 7 to 100 milliseconds. So we're talking about a pretty tiny event here. So for you to actually perform any sort of work, to actually move any sort of load, you're going to have a sustained muscular contraction where you've got a repeated stimulus. So I want to go ahead and show you a couple of terms here on this twitch. Again, think about this. We're talking about a single fiber, not a muscle body. When you stimulate a muscle to twitch, there is going to be what they refer to as a latent period. That's giving time for the calcium to be released from the sarcoplasmic reticulum, time for that action potential to run down the T-tubules into that muscle cell, for all these fibers to start to actually have the cross bridges forming. So then, after the cross bridge formation, you've got the actual contraction period. This is where your myosin heads are flexing. That's your power stroke. And then you've got the relaxation period. Relaxation period, now you've got the breaking of the cross bridges, and you've got the muscle returning to the original resting length. So it's important to recognize this is a single twitch because of some definitions I'm going to be giving you on the next few slides. Let's talk about the pattern of the stimuli as far as how it relates to when it occurs during that phase of the twitch. So I'm going to draw this over to the side here, and then we'll look at the chart. What you're seeing on this particular top chart is you're seeing stimuli arrive after the muscle fiber has gone through the complete stage of relaxation. So remember, you've got your resting muscle, you've got your contraction, you've got your relaxation, and now you're ready for another stimulus and another twitch. However, what happens if you have your twitch, you've got your relaxation starting, but before you get to the bottom of that, you apply another stimulus. Then what happens is you build up a little bit higher. Now let's say we do another stimulus, and it's going to come up again even higher. So when you're stimulating the muscle before you've gone through the complete relaxation phase, it's going to add up. So it's going to end up looking like that. So this is a very strong buildup of force or tension in the muscle. So here's another diagram. Again, what's happening, they're applying the tension before the muscle fiber goes through the stage of relaxation, before it completes relaxation. So it builds up over time. This is going to give you a couple of different patterns here. Now, again, we're talking about, everything we're talking about here is wave summation, but wave summation can lead to a pattern called incomplete tetanus, where basically the stimuli are coming so quickly that the relaxation phase is, in fact, the entire cycle is really shortened. Incomplete tetanus is the type of tension that we use to perform most of the work that we do in our everyday life. Now, what happens if the stimuli are coming even faster and faster and faster? It can build up to where this smooths out and forms what they call a complete tetanus. Complete tetanus is not sustainable for a prolonged period of time at all, because you're going to end up totally depleting calcium and depleting the ATP supply. But what happens here is that you never, ever enter the relaxation cycle. So over here, you've got all the phases. They're just really shortened. Over here, there's never a relaxation. So what's happening here is the calcium is simply staying inside the sarcoplasm, and it's continuing to keep on cycling those cross bridges. Again, this is not going to be a sustainable amount of tension that you're producing. So during your complete tetanus, pretty much what's happening here is all the cross bridges that can be formed are forming. Now, I'm sure you've heard the word tetanus before, and you're thinking about the disease process, tetanus. There's actually a bacterium called Clostridium tetani, and the toxins from the bacteria causes this rigid paralysis that is known as tetanus. But don't get confused with that just because the disease is called that. What we're talking about here is a normal physiologic phenomena. But again, remember that this is not going to be a very sustainable way to produce force. So wave summation and incomplete tetanus, and even complete tetanus, those are things that you're going to be seeing in skeletal muscle. A different type of pattern that you'll see in cardiac muscle is called trepa formation. So what happens with trepa is you go through the entire cycle of relaxation before the stimulus produces another contraction. So this is going to give you a much slower pattern. This way you're not going to go into a incomplete or complete tetanus, which you wouldn't want to. You would not want that to happen to your heart muscle. So the wave summation gives you 4 times the strength of the trepa formation, but the trepa formation gives you a much more controllable type of force. So make sure you know that trepa formation is seen in cardiac. Now, I always had trouble with this in class because trepa is actually German for steps. And people think wave summation looks like steps, which it does. But so don't be confused. Trepa, I prefer to think of it as a stepping stone type thing. So you've got these level stepping stones. So now you do build up a maximum tension in trepa. If you see here, your initial contractions are about half the force of your later contractions. But again, you're not adding on from one contraction to the other like you see in wave summation. With trepa formation, you've got these individual contractions. The reason that's possible is because of what they call a plateau period during which a calcium stays in the sarcoplasm instead of going back into the sarcoplasmic reticulum. So no matter how hard you try, you cannot stimulate a new contraction before the relaxation phase is completed. So make sure you know the differences between wave summation and trepa. Remember, wave summation, the stimulus is being applied and resulting in a contraction before you go through the period of relaxation. So if it happens very rapidly that you're getting the stimuli, you can get what they call incomplete tetanus. If the stimuli are coming very, very rapidly, then you can actually get what they call fused tetanus. But with trepa formation, you go through the entire relaxation cycle before you respond to another stimulus. So remember, this is skeletal muscle and this is cardiac muscle. Let's take a look at some different types of muscle fibers. Don't worry about this chart. I just simply put this on here as an introduction. This gets into a little bit more detail than what we need to worry about. But you may have heard the term before, fast twitch fibers and slow twitch fibers, talking about people that are good at endurance versus weightlifting. So let's take a closer look at that. You're going to have to endure my bad artwork here because I wanted to take you through a couple of terms here that I've mentioned to you in previous chapters, but we're going to talk about in detail in 142 when we get to chapter 25, everybody's least favorite chapter about metabolism. And that's going to hopefully help you understand the terms when we start talking about the types of muscle fibers. So this is supposed to be a hamburger. So you eat the hamburger, and you're going to be breaking it down to little pieces with your teeth and the roof of your mouth, the arugula in your stomach. So it's now in your GI tract. You've broken it down into the individual amino acids and glucose and fatty acids, all the lipids. You're going to be absorbing it into your bloodstream. From there, it's going to go through the interstitial fluid into the cell. Now, what we're going to talk about right now is strictly going to be carbohydrate metabolism. So let me put my emblem up here for carbohydrate. So what happens when you bring the glucose, the carbohydrate, into the cell, I hate to get into too many details here, so don't get hung up on details. The first thing that's going to happen, you're going to end up slapping a phosphate on there to trap it inside the cell so it can't slip back out through the cell membrane. But it's going to undergo a process called glycolysis, glycolysis. You're going to break it down. So now you've broken it down and you've released a tiny bit of energy. So glycolysis is pretty quick, but it doesn't give you a whole lot of energy. But the good thing is you do not have to have oxygen. You don't have to have it. So it happens when oxygen is present. It also happens when oxygen is not present. But now let's say we're going to take one of these little mitochondria and blow it up. If you have plenty of oxygen, you're going to take the byproducts of fructose that comes out of glycolysis and you're going to put it into the mitochondria. So it's going to go through a couple of stages here in the mitochondria. It's going to go through what they call the Krebs cycle. And then the final result is going to go through what they refer to as the electron transport system or chain, depending on how you want to call it. So this is actually where most of your energy is produced, especially in the electron transport system. You get a tremendous amount of energy from this part right here. However, you have to have oxygen for it to happen. So this is referred to as oxidative respiration. I sort of say oxidative phosphorylation, and we'll get to that term in later chapters. So this is oxidative. It gives you a lot of energy. This is glycolysis. It does not give you very much energy, but it happens very quickly, and it does not need oxygen. So hopefully those terms, as we go through the next few slides, will help you understand that a little bit more. What they refer to as a slow twitch fiber is a slow oxidative fiber. That means you have to have oxygen. It will not work without oxygen. These are large fibers. It looks red due to the high number of mitochondria. Remember, mitochondria, the little power factories of the cell that we were just talking about. That's where the bulk of your ATP is put together, your energy. So these take longer to contract, but they can keep on working much longer. So these are going to be really brought into play for endurance people. So these are going to be your slow twitch, oxidative fibers, and again, they allow you to have more endurance. They take longer to contract, but once they get there, they can keep going for a longer period of time. The type 2 fibers, you actually have two types, what they call a fast oxidative glycolytic, which is kind of a medium in the middle of the two terms here, oxidative versus glycolytic. So you've got a combination here. These are sort of the mid-sized, and it depends a lot on glycolysis and to some degree on the mitochondria using the oxygen. So there's not nearly as many mitochondria, so this is going to be more white in appearance. If you think about a chicken, think about light meat and dark meat, or some people say white meat and red meat. So the chicken breast, where the wings are attached to, is white meat. A chicken can fly, but not very fast, not very far. That's why they don't have to worry about putting tops on chicken cages or chicken coops, because it's really difficult for them to escape. However, a chicken walks around all day long. Again, not very fast. You can catch one if you put any serious effort into it. But so the legs of the chicken are dark meat or red meat because they're using a lot of that slow oxidative respiration. I'm sitting here on a mountaintop recording this, which is why I'm having to give you some really bad artwork instead of being able to depend on snatching a picture off the internet. You've had to listen to my dog snore through some of my productions here. She is not happy with bad weather, so you're going to be hearing a dog pant in the background because she's a little bit nervous. We're getting a storm here, so bear with me. We've progressed from snoring to hassling. So the second type of the type 2 fibers, referred to as 2B, don't worry about these numbers 1 and 2A and 2B. Worry more about the actual terms themselves. This is the ultimate fast twitch fiber here, fast glycolytic. Again, white meat. This is anaerobic. Now, the term anaerobic, you've seen that before, but you may not remember. That's from the earlier chapters. Anaerobic means you do not have to have oxygen. So for the people who are doing the sprinting, they're going to have a higher proportion of the fast glycolytic fibers. So again, this is not depending on oxidation. This is not depending on mitochondria. So you can run very quickly, but you're not going to run as far. I always use an example here about thoroughbreds versus quarter horses. If you were to put a quarter horse and a thoroughbred at the starting line and fire the starting pistol and they're off in a race, what's going to happen is initially that quarter horse is going to be faster than that thoroughbred. You're going to have this big burst of speed, and then after about a quarter mile or so, that quarter horse is going to fatigue and start to slow down, but that thoroughbred is going to keep running pretty well for about the next mile, mile and a half. So that's where the term quarter horse actually comes from. They have this lightning burst of speed, but then they fatigue very quickly. So our quarter horse is going to have a lot more fast glycolytic fibers, and the thoroughbred is going to have a lot more of these slow oxidative fibers. I want to give you a definition here of a motor unit because we'll be talking about motor units and tension in the upcoming section. A motor unit is the nerve cell or the neuron and all the muscle fibers that it controls. So it's always one neuron and a variable number of muscle fibers. If you have a motor unit that controls your eyelid is going to be one neuron and a small number of muscle fibers. You've got a very fine control. If you have an area of, let's say, greater force, but less fine motor control, let's say in your hamstring muscles or your gluteus maximus, one of these large muscles but little fine-tuned, fine motor control, that's going to have a large motor unit where you're going to have one neuron and perhaps thousands of muscle fibers to go with it. So understand that a small motor unit will give you much finer control than a large motor unit will. Understand that it's always simply one neuron and then a variable number of muscle fibers. So let's get the definition here of recruitment. Recruitment literally means how many motor units are you going to recruit? How many motor units do you need to perform an action? So if you're going to be performing an action that's going to take a long time, then what's going to happen, you're going to actually rotate the motor units so you don't fatigue them. In other words, very few times we'd be actually using all the motor units in a given muscle. Let's say that you're going to be picking up a bucket full of water and it's heavy and you're having to hold it for a sustained period of time. Your arm is going to be spontaneously, let's say you're going to be using your flexor carpi radialis and your flexor carpi ulnaris, those flexor muscles, picking up this bucket. You're not going to be using all motor units. You're going to be rotating them through so your arm does not fatigue as quickly. Now, eventually it will fatigue, but that process of recruitment is going to help rotate. In your postural muscles, there are some units that are always active, and that's how you maintain some degree of posture. You're not rigid, but you've got some muscle units that are always going to be turned on, basically. So if you're going to pick up an 8-ounce object, you're going to recruit much fewer motor units than if you're going to pick up an object that is 8 pounds. Recruitment can also be talked about when you're talking about different types of muscle fibers. We talked about your glycolytic fibers and your oxidative fibers. You're not going to have any muscles that are all one or the other. All your muscles are combinations of those types of fibers. Now, genetically speaking, you might have more of one type of fiber than your friend. You might be better at endurance than they are, or you might be better at very fast, explosive type movements. You might make a good sprinter compared to your friend if you have a higher proportion of those types of fibers. But everybody has a combination of fibers. So if you're going to, let's say, pick up something that's of medium weight, let's say you're going to pick up something that's about 5 to 8 pounds here, your slow twitch fibers are the ones that are recruited first. Now let's say you're going to be picking up something that's going to be 30 pounds, takes more effort. Your slow twitch fibers get recruited first, and then as you put more pressure trying to lift that load, your fast twitch fibers get recruited. So the fast twitch, the type 2B, are going to be recruited at maximum effort. So again, you're going to be calling in these different types of muscles at different types of strenuous activity. And it makes sense if you think about it. If you're picking up something that's fairly lightweight, let's say you're picking up something that's maybe a pound or two, you can carry that for a long ways. Let's say you're going on a walk for a few miles and you're carrying a water bottle in your hand. The slow twitch fibers that have a lot of mitochondria, they're using a lot of oxygen to allow you to be fairly efficient at producing ATP. You're not going to really fatigue and drop your water bottle from exhaustion. If it were flipped, if you were having to use your fast twitch fibers first, you would actually run out of energy and end up dropping your bottle just a short ways into the walk. So the first fibers you call into play are going to be the slow twitch fibers, the oxidative fibers. And it's only when you really are trying to do something more strenuous that you start calling in the glycolytic fibers. Okay, I wanted to throw this slide in there, simply giving you an idea, going back and combining a few concepts here. we talked about motor units. Remember, a definition of a motor unit is one motor neuron or one nerve cell and all the muscle fibers that it controls. So let's say that this is representing a fascicle, and here's a motor unit going to a motor neuron over here. So here you have a large motor unit giving you more force, but less fine motor control. Here you've got a small motor unit. You've got a neuron over here, but you're only innervating a few fibers here, so you've got fine motor control there, but you can't produce a lot of force. So the other thing to point out on here is these three different types of fibers. You've got your large, your type 1, your slow oxidative fibers. You've got your 2B fibers, these mid-size, and then you've got the small representing the type 2A fibers. So you've got all three fiber types. This is what this is telling you, is that your skeletal muscles have all three different types of fibers, and you have all different sizes of motor units combined. That allows you to use a minimal amount of force for a minimum amount of a job, a minimum amount of a load. If you're going to require more load, more tension to overcome that load, then you can simply recruit more motor units, and you can sustain that motion or that movement for a longer period of time as you switch from your slow oxidative to the fast glycolytic fibers. This chart might give you a better idea. So here is when your activity is first starting. So you're calling in your first motor units. These are going to be your slow oxidative fibers. You're continuing to perform the activity. You're continuing to have to overcome that load. You're going to recruit the next motor unit. So as you maintain that force, you're going to be calling in your fast oxidative glycolytic fibers. Remember, those medium fibers kind of in the middle. And then as you get to a longer period of time, you're starting to really fatigue all the earlier fibers, you're going to start calling in your fast glycolytic. So think about the fact, again, you might be able to pick up a 5-pound weight and hold it for a long period of time. But think about the weightlifters that you've seen in the competitions in the Olympics or something like that. They might be trying to lift 200 pounds. So they bend down, they pick up the barbell, and with a sudden explosive quick action, they jerk it up and force it over their heads. But they can only maintain that for just a small number of seconds. So they're using that fast glycolytic fiber group, but again, it's so short-lived because you don't have that oxygen, you don't have that extra ATP production. So that's sort of your snatch and grab, and that's what you're looking at when you start looking at this maximum effort over here. Over here, maximum force, short period of time. Over here, we had low force, longer period of time. Now, I told you at the beginning of the lecture that muscle contraction is active and relaxation is passive. In other words, you relax by simply stopping contraction. So what returns your muscle then to the original length is going to be a combination of three different things. You need to know these. First off, the elastic forces of the connective tissue. So remember we talked about tissue types in chapter 4. We talked about in connective tissue, the main fiber types. You have elastic fibers and you have collagen fibers. Those elastic fibers, they're protein fibers and they're crimped. And so when you stretch them out, they're going to recoil. So when you perform an action with a muscle, you form a contraction, you relax that muscle, it's going to go ahead and relax back out. So the opposing muscle contractions. Now, we haven't really talked about agonist and antagonistic muscles yet, but I'll give you an example that you're probably familiar with, your biceps and your triceps. So to contract your biceps, you have to relax the triceps. When you relax your biceps, your triceps are going to contract. So you're going to have your opposing muscle contractions. And then the other thing is gravity. While you're sitting there listening at this, go ahead and flex at the elbow, contract your biceps, and then drop your arm. And think about the fact that as you drop your arm, gravity is pulling your arm downward. So gravity is a main force here. So those are the three things that return a muscle to the original resting shape and length. Now, back on my artwork with a hamburger, we were talking about glycolysis and metabolism a little bit. Now, again, this is not something that you need to learn these numbers, but I just wanted to give you some numbers, give you some idea of the magnitude of the quantity of the ATP you have to have for sustained muscle contraction. Each fiber, each muscle cell has 15 billion thick filaments, the myosin filaments. So each filament uses 2,500, so you can do the math. So 15 billion times 2,500 molecules of ATP will tell you how much ATP one muscle cell is going to need. So the muscle cell is going to have enough ATP to begin and also to maintain a contraction. And we'll talk about muscle fatigue in just a moment, when you actually run out of the ATP and you cannot continue on. When you're resting, your muscle is actually creating ATP at a level that's more than what you need. And so the muscle tries to be efficient by storing the extra energy. And it stores it by transferring a phosphate to creatine. And it results in a product called creatine phosphokinase. Now, this is the same thing as creatinine phosphokinase. That was a term that I was taught to use, but evidently things have changed, and I would say creatine. So basically, I put this in here to show you this term creatine phosphokinase. That is an enzyme that is used to break this back apart when your body is needing to release that extra energy. So if you, for example, let's say you run a marathon and you have blood work done the day after, you're going to see a high level of what they call CPK, or creatine phosphokinase, because the creatine phosphokinase was needed to break this down to release the energy in those bonds. After about a half hour of sustained exercise, your body will have depleted the storage of energy that it had saved up in the creatine phosphate, and your body's going to have to find some other form, or some other supply, I should say, of ATP. That's when the glycolysis and the oxidative metabolism is going to come into play that we mentioned earlier. I probably should have given you these terms earlier. I mentioned them, but I didn't really have them written down anywhere for you. You actually saw these terms, I believe it was in chapter 3, but let's go ahead and give them again. When I was talking earlier about making energy and using the mitochondria, you had to have oxygen, and that's referred to as aerobic metabolism. So you have to have oxygen for this to happen. It's very efficient. It gives you 34 molecules, actually 34 to 36, depending on the book you read, of ATP for every molecule of glucose that your body's breaking down. So anaerobic metabolism, you do not have to have oxygen, but it's very inefficient. You only get 2 molecules of ATP for every molecule of glucose you break down. So the other thing that happens when you're going through anaerobic metabolism is a byproduct is lactic acid. So you end up creating what they call pyruvate or pyruvic acid. That converts almost instantaneously into lactic acid, and lactic acid can build up in your muscles, and that causes muscle soreness. That can actually cause muscle destruction if it's on high enough of a level. So after you've had a chance to rest and recuperate, that's lactic acid is going to be reconverted, process of gluconeogenesis, don't worry about that word, we'll get it later, in a process known as a Cori cycle, which happens in the liver. So just to kind of give you a rundown that we've already talked about before, but you eat something, you're going to be breaking it down into glucose and lipids and fatty acids and amino acids. Your body really prefers to digest glucose. It's the easiest source of energy it can get. So this glucose is carried into the cell. It undergoes glycolysis first, and then the products of glycolysis are carried into the mitochondria. And remember, the mitochondria is where your Krebs cycle and your electron transport system occur. So for this to happen, this is your aerobic, and you have to have oxygen. If anything happens and you do not have oxygen, then none of this can happen, and you're stuck in glycolysis. So while you might have a little bit going over here, the bulk of your glucose metabolism is not in glycolysis, and you're not very efficient. You're only getting that 2 molecules of ATP for every molecule of glucose, and you're accumulating that lactic acid. So this is not going to be very sustainable for a long period of time. Picture being on a treadmill, and you've got to set at a medium level for walking. And you're walking about 3 miles an hour. You're comfortable. You're talking to the person on the treadmill next to you. You might be able to go for several hours this way. You're working at an aerobic level. You're getting plenty of oxygen. You're not having any trouble getting your breath. And your body is continuously taking the glucose and converting it into a high level of ATP. Now, let's say somebody reaches over and hits a button on your treadmill and cranks it up to about 5 miles an hour. Now you're running. So what happens, you can't breathe fast enough to get enough oxygen for this side over here to happen. So again, you can't go there, so you're stuck on this side. So you're stuck in glycolysis. And because you're only making 2 molecules of ATP, you cannot go very long. Me personally, I wouldn't make it to the parking lot if I had to run from here to the car, but I could walk from here to Manassas. So over here, you can only go for a very short period of time. Over here, you might go for a long period of time because, again, you're making 34 to 36 molecules of ATP. Over here, in glycolysis, you're accumulating lactic acid that's going to build up and potentially cause muscle damage. So let's say you're on that treadmill and you've been jogging along there pretty briskly. Eventually, again, you're going to be stuck in more of a glycolytic state than you are an oxidative state. You're going to be running out of your ATP. You're also going to run out of calcium, and you're going to end up in what they call muscle fatigue. You're going to accumulate lactic acid. Small amount of lactic acid you might not be aware of, but I'm sure at some point in your life you've done enough exercise that the next day your muscles are sore when you went to move. So that was actually damage to your muscle fibers from lactic acid accumulation. So you can actually continue to have damage occur for up to 3 days after the initial exercise. They actually have a syndrome called delayed onset muscular damage. And you can actually break your muscle proteins down so much that you excrete it in your urine. The urine can be very dark, and those protein molecules, very globular, as myoglobin being broken down, they can actually stop up the tubules in your kidneys and result in kidney failure. So you may have seen someone at the very end of, let's say, a marathon. They have just enough energy to get to the finish line, and they absolutely just collapse on the ground. So if they're in dire straits, one of the first things they'll do is hook up an IV fluid solution to them to try to make sure they have enough fluid volume in their blood vessels, but also to keep on trying to flush any lactic acid and muscle damage byproducts out of those kidney tubules. So in every class I have, at least my face-to-face classes, I always have a few people that are interested in muscle physiology as far as exercise training, personal training, that sort of thing. So they always have questions about strength training. When you're actually going through a exercise program, you're becoming stronger and you're becoming more able to perform certain tasks without thinking about it. We've talked about that a little bit before, but you may have heard the term muscle memory, and this is where we're going with this. When you're exercising for a long period of time, you're going to affect the nervous system, and what's happening here is you're calling in more motor units. So you're increasing your recruitment. You're also decreasing the activity of the inhibitions of the nerve cells that will inhibit the motor units, so you can turn these motor units on a little bit quicker. And then you're also causing better coordination between the nerve cell and the muscle fibers. So these are all methods of increasing your ability to perform an exercise quicker at a higher rate and with more ease. Now, we also are going to affect the muscular side of that. So let's look at the next slide for that. Your muscles will adapt to increases in exercise programs by what they refer to as hypertrophy. Hypertrophy is talking about increase in size. So what's happening here, you're not forming new muscle cells, but you're adding structural proteins to the muscle fibers themselves. And this is referred to as hypertrophy. The opposite of that would be atrophy, which simply means that you're losing structural proteins and the muscle mass decreases. So after a few months of not exercising, you can actually undergo what they refer to as atrophy. So there's two types of exercises I'll mention here: endurance exercises, and that's when you're going to be really calling on those slow oxidative fibers, and they don't require a whole lot of force, but they involve a whole lot of repetitions. So what's going to happen here is you're going to be increasing the amount of capillary networks forming around the fibers. The term for that is angiogenesis, just formation of new blood supply, and it improves the ability of the body to get oxygen to those muscle fibers. Resistance exercises will actually use a lot more of the fast glycolytic fibers and will produce high forces for short periods of time. And that's going to give you more of the bulky muscular definition that you see with a bodybuilder, as opposed to the long, leaner endurance runner. So just remember that with exercise, what you're trying to do is increase your size, not the number of fibers. And if you're doing endurance training, you're going to be training for a marathon. That's going to be your slow oxidative. So you're going a slow rate for a long period of time. That might be your low weight, high rep type exercise. If you're doing the weightlifting, the bodybuilding, then you're going to be doing the resistance exercise. And that's going to be the high weight, low rep. This is going to be more of your fast glycolytic or even your fast oxidative glycolytic fibers. People who are in serious competitive form here sometimes will actually try to get a competitive edge by using different types of performance-enhancing medications. These are illegal, and they can also be pretty dangerous in results. There is a medication called EPO, erythropoietin. It's a hormone that's normally produced in the kidneys to increase the number of red blood cells that you have circulating to carry more oxygen. They formulated this to help people with severe disease such as cancer, that sort of thing, to give them more energy. And you'll find, in fact, this is one of the drugs that Lance Armstrong was busted for using in his long-distance cycling. Again, the whole idea was to be able to add to their endurance. You'll actually see a lot of anabolic steroid use, and the idea there is to increase muscle mass. And the problem is that has a very dangerous side effect on the heart and blood pressure, as well as nervous system function. You've heard of roid rage, and there have been many number of murders. A lot of the bodybuilders, the wrestlers, that would be taking the anabolic steroids, and they would come home and beat their wives and children to death, and they blamed it on the steroids that they were using. Another, and this is actually something you see over the counter, it's not an illegal substance, but it's a creatine supplements. And again, the idea is, I told you before, that your body will actually store energy, because what it's doing is taking the phosphate and it's going to be adding it to the creatine. So it's storing energy in this creatine phosphate combination. So the idea is if you add creatine to the body, you might be able to store more energy to give you that really strong initial burst of energy. They just, from a scientific basis, they really haven't proven a whole lot here, and I'm a little bit concerned about effects on things like kidneys. Let's take a brief look at cardiac muscle. Again, we're going to keep the bulk of our discussion here for chapter 20, but there are some things you do need to know about cardiac muscle. Remember that it is striated, appearing striped. Make sure you remember that it is involuntary. You have no control over it. Now, you might say, well, what about relaxation procedures and biofeedback and all this business? Well, you're affecting your nervous system, which has a secondary effect on your heart rate. So that's not really a direct control. It's still involuntary. It requires a tremendous amount of oxygen. So it is a very aerobic muscle. It has a high number of mitochondria in those muscle cells. And this is one of the reasons that smoking is such a dangerous habit. Because you're causing lung damage, and you're also causing arterial damage, so you're making your heart beat that much harder. At the same time, you're starving your heart of oxygen. So your heart cannot perform nearly as well, and that's one of the ways that smoking can lead up to increasing the likelihood of a heart attack. So the cell junction, we've talked about these before, intercalated disc. There's simply a way that your cells are held together that allows a very simultaneous contraction, so your heart muscle cells are all beating at one time, not quivering like a little bag of worms, which that's not good. That would actually be a fibrillation, which is not compatible with life. It also allows the cells to hold together because the contraction of the heart is actually pretty forceful. So these cells are not multinucleate. They only have a single nucleus, unlike the skeletal muscles. You don't have the terminal cisterna. You have water T-tubules. Remember, they use TREPA. They don't use a wave formation. And that happens because, remember, the calcium ions, instead of being pumped back into the sarcoplasmic reticulum, they actually stay in the sarcoplasm longer, producing a plateau effect. So you've got a longer refractory period, which means you cannot make that muscle fiber contract any faster than it's going to. You go through a complete relaxation phase each time. So what you need to know test-wise, again, cardiac muscle, it's involuntary, it's aerobic, it has intercalated disc, and it uses TREPA. So those are what you need to focus on for testing. One more thing I want to say about cardiac tissue at this point is make sure that you realize that the heart has what they refer to as pacemaker cells. And that means these cells are capable of producing their own action potential. So that means that the heart is capable of setting its own rhythm. So the term for that would be autorhythmicity. Smooth muscle cells, remember these are involuntary also, and they're also non-striated, which is why they say smooth. They're spindle-shaped cells. They also have pacemaker cells. You'll find these, for example, in your GI tract, in your gut, deciding when to push that sausage biscuit a little bit further down. So they do not have the trypamyosin. They have something called calmodulin. They do have actin and myosin, the thick and thin filaments. They're bundled together. Instead of talking about motor units, they talk about single units and multi-units. So you can have a single cell contract, or you can have an entire sheet or a bundle contract. On these next couple of slides, I'll be pointing out to you what you need to know about smooth muscle for the exam. Again, the big thing here, make sure you know that it's involuntary and non-striated. Make sure you know the examples of where you will find smooth muscle. You're talking about lining the blood vessels, and also the hollow organs. So I'm talking about lungs, I'm talking about, let's put down here, respiratory tract, GI tract, reproductive tract, urinary tract. Those are all smooth muscle wrapped. Relatively speaking, these are fairly small cells. They're single nuclei. These cells are designed not to give you a large amount of force. They're designed to give you a continual amount of force at all times. So these cells have what they refer to as a resting tone. So for example, when you're not actually eating or swallowing, your esophagus is still maintaining a resting motor tone. It's still contracted. Otherwise, you'd be aspirating air every time you breathed in. So again, this is for a long, sustained, low force type of activity. This concludes the chapter 10 section of this, and we're moving into the individual muscles in this next section. Chapter 11, talking about the individual muscles. The initial part, there are some things I want to point out, and then the bulk of the chapter is actually learning to identify muscles. And at the end of this PowerPoint, I'm going to be giving you some brief list of the muscles you need to focus on for the final practical exam. Muscles can be described by the arrangement of the fascicles. So what we're talking about, remember, a fascicle is a bundle of muscle fibers. If you've ever cut a piece of meat, a steak, you've been looking at the fascicles. Those are you talk about the grain of the meat, you're looking at the fascicles. So if the fascicles are arranged parallel to the long axis, it's referred to as a parallel muscle type. So what I want you to know here for your test, recognize the examples that I give you. So I'll be giving you some matching questions or some multiple choice questions. And if I say, give me an example of a parallel muscle type, you say, the biceps brachii. The word convergent means coming from a broad area to a small area. So a convergent muscle, an example here is going to be the pectoralis muscle. So this muscle right here, you see how it comes from a broad area down to a smaller point of attachment. So I'm going to point out the word aponeurosis here. An aponeurosis is simply an attachment it's like a tendon that's been ironed out into a flat sheet. So anytime you see this broad white sheet on a muscle model, that's referred to as an aponeurosis. It's still tendinous type material. A raphe simply means a line of tendinous material. The word pinnate refers to a feather. So if you were to picture a feather like that, that's actually very much what a bipinnate muscle would look like. So your bipinnate has both sides here, and your example is going to be the rectus femoris muscle. What you're looking at is right there. So there's your line through the middle, and then you can see the fibers coming down like that. So unipinnate would mean that you simply only have half that feather-like shape, and your example is going to be the extensor digitorum of the arm. And then a multipinnate is simply meaning that you've got attachments in which the muscle fibers wrap all the way around the tendon. So multipinnate example is going to be the deltoid muscle. That's if you picture the top of your shoulder, the top of your upper arm. The whole point of the multipinnate muscle is it allows you to change the direction of your force or your pull. Go ahead now and pick up your arm, raise your elbow to where it's level with your shoulder, and then you'll see you can actually move that arm in a nice circular motion. That's because of that deltoid muscle and the multipinnate attachment. So again, for testing purposes, simply be able to identify the type of muscle. It's going to be a matching or multiple choice type situation. This is always pretty easy for people. The circular muscles, they're going to be the sphincters. They have the ability to open and close a tube. So you have the orbicularis oris, which is around the mouth, oris for oral. Orbicularis ocularis, around the eye, oculi. And then the orbicularis ani, or anus. So that would be your external anal sphincter. So here is your orbicularis oculi. allows you to open and close your eye. Now, I don't do a whole lot here talking about classes of levers. Your book will talk about it. We've already told you that muscles and bones act together as levers. So the joint simply acts as the fulcrum. I do want you to know that most of the muscles in your body act as a third-degree lever, and that's your example here. Your joint being the fulcrum, your muscle is going to contract to be able to lift that load. So this is your third-class lever right here. Going back to your basic definitions, your muscle is going to be providing force to overcome resistance. That resistance is produced by the load. No muscle chapter is complete without mentioning origins and insertions. These always strike fear into the heart of most students. For testing purposes, I'm not going to ask you at this point to know origins and insertions, simply because it's going into detail that you're not going to be using enough to recall later. Depending on your line of study, this may be something that you have to know them all and know them in pretty well detail. But in the context of one semester A&P course, I think it's just gratuitous suffering for you. So origin of a muscle is the point of attachment. It's where it starts. So it's going to be proximal. Insertion is going to be the distal or the moving in. Now, in more recent muscle physiology courses, they're getting away from the terms origins and insertions and simply talking about proximal and distal points. The reason for that is there are some muscles that are actually much more confusing if you're starting to talk about origins and insertions because of the way they cross the joints, and it's easier just to say proximal and distal. I mentioned to you earlier about agonist and antagonistic muscles, and what I'm talking about there is the way that muscles interact with each other. So this again can be confusing because every now and then you'll find a muscle that is an agonist to one muscle and muscle pairing, but it's actually an antagonist from another muscle pairing. So I'm going to keep it simple here. An agonist is considered to be the primary mover or the prime mover of a muscle pair. An antagonist is going to be the one that opposes the agonist. So you cannot have an agonist and an antagonist contract at the same time. And again, I go back to using the example of the biceps and the triceps. When one contracts, the other must relax. In that particular pairing, the biceps is the agonist and the triceps is the antagonist. Now, when muscles are synergistic with each other, it means they're actually assisting each other. An example here would be the brachialis muscle assisting the biceps brachii. So when they're synergistic, they work together. So your synergist is a small muscle that would help an agonist. Although the more proper term there would be helping the prime mover. When you look at learning to name the muscles, it can be a really intimidating task. But if you get down a few basic terms, it's going to help you at least be pointed in the general direction. We can thank the early Greeks and Romans for giving us some of these terms. We've continued to name muscles using those root words. So if you learn the basic roots and suffixes first, you're going to be well on your way to kind of learning groups of muscles. So first off, if you look at the name of a muscle, you may be able to identify where it is based on the location. So for example, the temporalis muscle, you're going to know to be looking around the temporal bone. The occipitalis muscle, the same way. Origin insertion, genioglossus, if you knew that genio meant chin and glossus meant tongue, then you would know the general area to look for that muscle. We've already talked about the arrangement of the fascicles. So here, rectus means straight, transversus means across, and then oblique would be an oblique like that. So your examples here would be your rectus abdominis. Let me draw an abdomen here so you've got your arms. Ooh, this is bad. So your abdominal muscles, your rectus abdominis will be going in that direction. Your transversus abdominis will be going in that direction. And then your obliques, your external and internal abdominal obliques, be going in that direction. So you've got layers. Relative position, intrinsic and extrinsic. I'll give you an example here, talking about the eyeball. The eyeball contains smooth muscle that allows you to dilate your pupil. So that would be the muscle of the iris. You also, and we'll see this in chapter 17, you have the lens that is held up by these little muscles called the ciliary muscles or ciliary body. And they will let you change the shape of the lens. So those muscles, the iris muscle and the ciliary muscles, those are all intrinsic muscles. Now, extrinsic muscles of the eye, they would be, for example, the muscles located on the outside of the eye, and you have all these rectus muscles that attach that let you move your eye upward and downward and obliquely. So extrinsic would be the muscles on the outside, intrinsic would be the muscles on the inside. Now also, you can take a clue from looking at the names having to do with the shapes and size of the muscle. Trapezius referring to trapezoid shape, deltoid referring to triangular shape. Size here, magnus, brevis, and major. It sounds like something from Starbucks. But brevis would be short, magnus would be very large, major is still a large muscle. Also, this will tell you your adductor muscles. If you adduct something, think back to some earlier terms, you're bringing it closer to the midline of the body, you're adducting. If you're abducting with a B, you're going to be taking it away. So think for a minute, if you're going to adduct, ADD, a leg, where would that muscle be? There's a muscle called the adductor magnus that lets you bring your muscle to the inside, closer to the midline. So that muscle is going to be on the inside of your upper thigh. Remember, a muscle performs by contracting, is shortening. So when you contract your adductor magnus, you're actually bringing your leg, you're adducting toward the midline. Also, an important concept to get here is going to be talking about your flexors versus your extensors. If you're going to be extending your fingers, you're going to be contracting the muscles that are on the top of your arm, on the dorsum of your arm. Think about anatomic position. That would be your posterior muscles. If you're going to be flexing your fingers, flexing your arm, you're going to be flexing the muscles on the ventrum of your arm. If you think about extending your toes or flexing your toes or extending your leg or flexing your leg, think about where those muscles are going to be located. So that will tell you if you're going to be looking for an extensor, you're going to be looking for something that's going to be on the top side. If you're looking for a flexor, you're going to be looking for something that's actually on the bottom side. Okay, so let's go through some muscles that you're going to be expected to know, both for the written exam and the practical exam. I've already told you that the extrinsic eye muscles for the eye are on the outside of the globe. You don't have to know them individually, just know that they're extrinsic. And the intrinsic eye muscles are inside the eyeball itself. The muscles of facial expressions you need to know. First off is going to be the buccinator. The buccinator is located right here. The next one is going to be on this particular slide, the orbicularis oris. Remember, the orbicularis simply means it's a circular sphincter muscle. The oris is going to be around the mouth. And then the orbicularis oculi is around the eye. The zygomaticus, if you remember the zygomatic bone is your cheekbone, then you have the two heads of the zygomaticus right here. Don't worry, as far as I'm concerned, about knowing which head is which. If I label that, simply tell me it's the zygomaticus. The frontalis, this is pretty easy. Look for the frontal bone. There's our temporalis muscle. The one thing that I'll go ahead and point out on this slide is you need to know the masseter muscle. It's the big one that lies right on top of the buccinator muscle. So go ahead and add masseter to your list. I've tried to give you a little bit of a cheat sheet. If you go through this PowerPoint and write down the names of all the muscles that I've isolated in the title, those are the ones I will be focusing on. So the muscles that help you chew, hopefully you know that's what mastication means. Here is a picture of the masseter muscle with the buccinator not being shown up under there, but remember masseter is massive. Your pterygoids are up between your ear area and your cheek or your jaw. So here's the medial and lateral pterygoids. Again, don't worry about medial and lateral. The temporalis muscle also is going to be helping you chew. Remember the T and J joint, so that this is your temporalis muscle. Just look for the temporal bone. The muscles I want to point out for the neck, number one is going to be the platysma muscle, which is a very flat, thin sheet of muscle. It's typically shown being cut and pulled downward. So this is what it looks like when it's not. So this is a platysma muscle, very thin. When you start to get a little age and you have that little turkey neck thing coming down under your chin, that's your platysma muscle. It sort of splits and hangs down when you get into the age that I am now. So let's see, one more to point out would be the sternocleidomastoid muscle. So that's this muscle right here. So the sternocleidomastoid, again, don't worry about the individual heads. If you decipher this word, sterno for sternum, so it starts with the sternum. Cleido means clavicle, so that's your collarbone. And then the mastoid for the mastoid process, if you recall, that's going to be located up here on the temporal bone. So make sure you can locate the sternocleidomastoid muscle. The omohyoid muscle, the hyoid bone, remember the little horseshoe-shaped muscle? That is a horseshoe-shaped bone. That's the only bone in the body that does not articulate. Anytime you see anything about the hyoid bone, you should know that that means the muscle is going to be located somewhere around the base of the tongue. So remember, the hyoid bone is the attachment for the tongue muscle. So it's going to be deep up under this mass of external muscles here. I'm probably not going to ask you on a practical to locate the scalenes because they tend to be deep to other muscles. But the scalene muscles are important. I point them out because they are called the accessory muscles for respiration. So they're going to be right here. So I just wanted to point those out because when we get to chapter 23 next semester, we'll be talking about the accessory respiratory muscles. So they're going to be helping elevate the clavicle to increase the internal diameter of the thoracic cavity to help you breathe. So again, there's your sternocleidomastoid muscle. And from this angle, you're starting to see a piece of the trapezius. So you get a much better view of the trapezius from the back, but it's really important that you learn to localize these muscles from all three angles. It's very difficult, if not impossible, for you to learn muscles simply from looking at a 2D representation on paper. I've had very good students that would miss something on a practical, on a model, because they were studying it on a 2D representation instead of a 3. And the one that I've got in mind right now, I remember, I'll never forget her saying, she said, "Oh, I didn't know that the triceps are wrapped around to the side like that." So be careful using 2D going to 3D. I'm not going to be using anything that I see here, except maybe the sternocleidomastoid muscle. I just wanted to point out, here's your hyoid bone, and there are all the muscles that are going to be attaching to. Remember, the hyoid is going to be your tongue attachment. So there's another view of your scalenes peeking out right there. Some of these muscles, although you don't get a good view right now of the levator scapulae, but you can look at some of these by their names and tell what they do. Levator scapulae is going to let you shrug your shoulders. So you get the point of it right there. It's going to be extending back, attaching to the scapula. These muscles, most of them, are actually part of your rotator cuff. So make sure you know these and know which one is not part of the rotator cuff. The supraspinatus and the infraspinatus. They're differentiated by the spine of the scapula. So if you look at the scapula without the muscles on it, you'll see that you have the uh, spinous process of the scapula. You have the fossa under it and the fossa above it, and when you put the muscles on top, it gives you the supraspinatus and the infraspinatus. On the other side, if you were looking at the underside of that scapula, you would see the subscapularis. And then you have the teres minor and the teres major. Here's the teres minor right here. And that is the fourth part of the rotator cuff. The teres major is not part of the rotator cuff. So your rotator cuff muscles are your supraspinatus, infraspinatus, subscapularis, and teres minor. So make sure you know those. But you need to be able to localize all five of these muscles on models and diagrams for the exams. So here again is your list, and this picture is showing the ventral side of the scapula, showing your subscapularis. Looking at the upper back and the back side of the neck, there's the rest of the trapezius muscle that I was referring to. You see sort of the trapezoid shape there. It's covering up the rhomboids. You have rhomboids major and minor. These are going to actually be covered, and I'm not going to have you identify those on the practical, but I do want you to know that if I say where are the rhomboids located, you're going to tell me they're on the back or on the spine. So levator scapulae, I mentioned that earlier. It's coming from around the front and inserting on the scapula, and so it's going to let you shrug your shoulders. Now again, this is a group of muscles. Most of these are not going to be on your practical. There's a sternocleidomastoid that is going to be on the practical. Trapezius will be on there. But I just wanted to show you the splenius capitis and splenius cervicis. These attach to the head and neck, and they help you rotate the head, flex the neck. So the longissimus muscle normally helps you hold your head up. Those big muscles in the back. Again, these muscles that I'm putting checks, these will not be on your practical. Okay, looking from the front, looking at the chest now, make sure you can localize the pectoral muscles. Make sure you can localize your deltoid muscle. The serratus anterior muscle, you have a serratus anterior and posterior, you need to know both of those. But the anterior, so for me, you see from the front, it's sort of interlaced with the external abdominal obliques. So I've had people mislabel those as the oblique muscles. So make sure that you can identify all of these. Those will be on the practical. Let me see, I missed the latissimus dorsi muscle. You really can't see much of it here. If you're into being a gym rat, people refer to those as your lats. But you get a peek of it there, you'll see that better from the back side. Again, simply using a different diagram, here's your big deltoid muscle, your pectoralis muscle, you're seeing a bit of the trapezius muscle, your sternocleidomastoid muscle, your external abdominal obliques on this side, and then your serratus anterior muscles over here. And again, there's peeking through the latissimus dorsi from the very back. So from the other things you can see here are going to be your biceps brachii and then your triceps brachii. You need to know basically all of the abdominal muscles, and this is pretty easy. So you have this big aponeurosis that's been cut away, so you can start to see these layers. So if all these muscles were not cut away, then you'd actually just have this big white sheath coming down. So when you start removing these, kind of dissecting down, let's go through what we're going to be looking at. Again, here are the serratus anterior muscles. There's the front side of the latissimus dorsi. These muscles are your external abdominal obliques. So when you pull those away, you're going to see the rectus abdominis. Remember, the word rectus means straight. So these are the muscles that will come straight down here. People talk about the six packs. That's what they're talking about. We all have six packs. It's just that some of us have more padding. I know I've got a healthy sheet of insulation on mine, so you really can't see it. But we all have our six packs. Okay, the internal abdominal obliques, they go the opposite direction than the external. So here's your externals going in that direction. When you cut down, you find your internals going in that direction. And then finally, your transversus abdominis is going to be your deepest layer. And remember, transverse section, that means these muscle fibers will be going in that direction. It's just that you can't see them when the top layers have been not cut away. So transversus abdominis on the deepest aspect, then your rectus abdominis, and then your internal and external obliques. Okay, muscles of the arm. If you're going to be in my face-to-face lab, this is actually a photograph of the model you're going to be using. So either way, you're going to have to know all of these muscles for your practical, be it online or face-to-face. So walking you through here, you're going to see the deltoid. Then you've got your biceps brachii. Always, always use the entire name of the muscle. There's a biceps femoris on the leg, so don't tell me just biceps, because I need to know the biceps what? Biceps brachii. Brachii means arm. So on the bottom side of the arm, then you have your triceps brachii. You have a little synergistic muscle down here called the brachioradialis, and if you break that name down, remember brachio means arm, so we're talking about upper arm, so it's going to insert on the upper arm. Originate from the upper arm and then insert on the radius. So that should help you find that little muscle right there. Coming down to the extensor carpi radialis longus. So it's an extensor, so you should know it's going to be on the dorsum of your arm, of your forearm. Let me see, extensor digitorum. It's going to help you extend your digits again, so you know it's going to be on the dorsum of your arm, and you should be able to trace this down to the tendons on your fingers. And then I'm trying to see what we can see in this view here. The flexor carpi ulnaris. Remember that your ulna resides under your pinky. So when you see a flexor muscle, you know it's flexor because it's on the ventrum of your arm, and it's going toward your pinky, then chances are high that it's going to be the flexor carpi ulnaris. And then the palmaris longus, when you trace that down, when you see the entire model or an entire picture, you'll actually be able to see that it's actually going down to your palm, so that should help there. So when you look for the radialis, it's going to your thumb. When you look for the ulnaris, it's going to your pinky. Same thing for the extensors. The extensors are going to be on the dorsum of your arm, on the back of your arm. The flexors are going to be on the ventrum of the under part of your arm. Simply the lateral view here for the practical, your deltoid, biceps brachii, the brachialis muscle, triceps brachii, then your brachioradialis is down up here, and on my iPad here I can't see what's down below. Don't worry about the ankylosing muscle. That's a muscle that's going to be assisting in the rotation at the elbow, but I'm not going to be using that on the practicals. So let's look at some of the back muscles. It's always interesting when you can start naming the muscles on a live model. I did have one student that told me that her way of learning muscles was to actually draw them onto her boyfriend. So if you happen to have a boyfriend that looks like this, then perhaps you can localize some of these muscles. And all power to you and happy learning. But alas, for the rest of us, we're going to have to learn it this way. So again, these are muscles that you're going to have to be labeling on an exam, or or identifying rather on a practical, depending on which from the course you're taking. So the trapezius muscle, again referred to as a trapezoid. Your teres major, you can see that sticking up under here. There's your deltoid again. What you're seeing, this little area here, it's got what they call the fascia overlying it, but you can actually see the infraspinatus sitting up under there. So let's see, the rhomboid you're not going to see on this picture because it's up under there, but your latissimus dorsi is going to be this muscle, this big sheet of muscle that you see. You can't see the bottom of it from this picture. Well, I changed the frame just a little bit, and I pulled it up, and there you actually see your rhomboid muscle right there peeking through there. It didn't show up on my earlier frame. Here's a better view, going a little bit further down. Again, there's your deltoid muscle. Here's your trapezius muscle. There you see one of the rhomboid majors sticking up. There you see your infraspinatus. There's your teres major. This big muscle, this big flat sheet of muscle, is your latissimus dorsi. You can start to see some of the abdominal obliques sticking out up under there. Now here's a diagram that's got one half of it cut away. So the posterior serratus muscles, you can actually see those being located right here. Again, you're not going to see them on most of the lab models because they're going to be covered up by this latissimus dorsi muscle. But if you look at this, they're going to be going the opposite direction as far as the anterior serratus muscles. So there's your dorsal edge of your external obliques. There's the edge of your internal obliques. Again, trapezius. There's your sternocleidomastoid going up to the mastoid process. Deltoid muscle. You're starting to see here the gluteal muscles. And then here, up, and because you've got, again, the cutaway, you can see some of the splenius muscles, the capitis, and the cervicis. Those are not going to be on your practical to label, but I just kind of wanted to point those out. There's your supraspinatus muscle. Again, there's the rhomboids. The erector spinae muscles, again, are not going to be on your practical. I just wanted to point them out. Just like the name sounds, it holds your spinal column erect. For testing purposes, basically just know where they're located. If I were to say, give you a multiple choice question and say which one of these would be visible if I remove the latissimus dorsi muscle and I had any of these first muscles listed, any of those would be the correct answer. And then obviously the gluteus muscles would not be correct because those would be located down lower. The gluteus muscles might be a good one to learn for your practical. Just another view of the arm using the lab model. You do need to know all of these for the written exam and the practical exam. So again, subscapularis, triceps brachii, flexor carpi ulnaris, trace it going to the pinky is going to help you locate it. And again, you're looking at the palm of your hand here, so you know that's going to be the flexor side. Your palmaris longus going to the palm of the hand, pretty simple on that one. Your flexor carpi radialis, you trace that as going up toward the thumb. So here's your brachioradialis. It's going to be wrapping over to the back side of the upper arm. Okay, here is the other side of your arm. So you're looking at it from the top. You're looking sort of at the dorsum of your arm. There's your deltoid. Your triceps brachii, your biceps brachii, your brachialis muscle, the brachioradialis muscle. The extensor digitorum, again, follow on down, and you'll see this is going to help you extend your fingers. Here is the small head referred to as extensor digitorum minimi. Don't worry about that. Just say, tell me extensor digitorum, and that's good enough. Extensor carpi ulnaris going toward the pinky, and your extensor carpi radialis is going to be over here going toward the thumb. Now I just put this in here just to show you a couple of things here. Remember the word pollex for thumb. So anytime you see the word pollicis, you have the flexor pollicis brevis and the adductor pollicis brevis. They're talking about the muscle that's going to be helping you bring the thumb inward or outward or to flex it. So remember, to abduct would be to move it inward. Adduct would be to move it inward, and then to abduct with a B would be to take it away. Digiti minimi, that's your pinky finger. So I'm not going to have any of these on the practical for you to label or on the written exam. I just wanted to show you these. And again, you should anytime recognize the word pollex for thumb to at least give you an idea where it is anyway. I want you to know the word lumbricals. I don't want you to know the individual muscles themselves, but the lumbricals are the muscles that you'll find between the metacarpal bones and the metatarsal bones. So you actually have lumbricals both in your hands and your feet. So they're between the long bones in your hand. Just pointing out a few things here, the retinaculum. So the retinaculum is going to be this sheet of tough fibrous connective tissue that helps hold the tendons down. So it's the retinaculum that they'll go in there and snip and enlarge when you have a carpal tunnel syndrome. This is flexor because it's on the ventrum of your hand. It would be the extensor retinaculum on the opposite side. So you also have one in the tarsal region. Again, it's keeping these tendons pressed down when you flex your hand. So make sure you know where the retinaculi are. So the only other muscles I was going to point out, I simply want you to know they're on the extremities as far as the hands and the feet. They're referred to as the dorsal interosseous muscles. So if you're looking at the back of your hand, the muscles on the dorsum between the metacarpal and metatarsal bones are called the interosseous. So on the palmar side, it's the lumbricals. On the dorsal side, it's going to be the interosseous muscles. And again, it's the same really for the feet and the hands. I'm not going to have you localize them on a practical, but I simply want you to know where they are. Moving our way down the legs, make sure you know the gluteus maximus. Everybody, I don't think I've ever had anybody miss that muscle. That's a pretty big one up here. So I'll often include that on the practical exam. There's a group of posterior thigh muscles referred to as the hamstrings, and it's composed of the semitendinosus, semimembranosus, and the biceps femoris. Now you will need to be able to localize all these muscles on the written and the practical exam. Biceps femoris, again, you have to tell me the whole word, don't just say biceps, you get it wrong, is going to be this muscle right here. Now, the semimembranosus is peeking through right here. The semitendinosus is this muscle right here. So the way that I always learned the semimembranosus muscle is the semimembranosus is medial, so M for medial. So it's hard to see on this diagram, I realize that. Find a better diagram online and look at the models if you're coming from face-to-face class. So there's your adductor magnus hidden up in there. Remember the adductor magnus, large muscle that pulls the leg inward, it adducts it. And then you're going to need to know the gastroc muscle, the gastrocnemius muscles. They're going to be your large calf muscles. That's your gastroc tendon inserting on your calcaneal bone. For my face-to-face class, here is your model. And again, looking at it from a lateral view, there's your gluteus maximus. There's your biceps femoris. It's much easier to see than what was on that previous diagram. That's going to be your semitendinosus. There's your gastroc. This muscle right here, the fibularis longus, also known as peroneus longus, you get it right no matter what you put. You can start to see the anterior tibialis muscle right there. Looking from the front, I haven't given you a good view from the front yet, but when you do, you'll be able to see the front leg muscle, front thigh muscle, is the rectus femoris. Remember, rectus meaning straight, femur telling me it's in the thigh. On each side of that, you've got some vastus muscles. You have a vastus lateralis that you see from this side. You have a vastus medialis that you see from the other side. And then you actually have a vastus intermedius that you don't see because it's buried up under there. Now, here is the head of the femur. So now you're looking at this from the medial side. So there's your adductor longus again. These two muscles are pretty easy. This muscle going down the inside of your thigh is referred to as a gracilis. Coming diagonally is the sartorius. Don't get those confused, as some people do. So again, there's your vastus medialis, and you're seeing the rectus femoris peeking over from the front. So again, there's your gastroc muscle. The group of the large muscles on the front part of the thigh are referred to as the quads or the quadriceps. So you're looking at your vastus lateralis, your vastus medialis, the vastus intermedius, which is actually again hidden up under here, and the rectus femoris. So make sure you can locate all of these on your practical exam. I'll point out a couple of things. The pectineal muscle right here and the iliopsoas, those will not be on an exam. I wanted to point them out. And I was going to point out the tentra fasciolata, which is going to be this band, this muscle belly, and this band coming right down here. And I'm pointing that out because a lot of people will say, oh, they have trouble with their IT band. It'll get sore with some athletic endeavors. And just wanted to point out that's where it comes from, the tentra fasciolata. The IT part of it comes from saying the iliotibial tract. So again, here's what I was talking about with the iliotibial tract. And then it runs into your tentra fasciolata, and there's the connective tissue band. Again, make sure you know the sartorius. Make sure you know the gracilis. And here is your rectus femoris. So that's the end of this chapter. I did not go any lower on the leg, but you do need to know the gastroc and the anterior tibialis muscles and the soleus muscles. You need to localize those for the practical. I've tried to isolate the major muscles. There's a lot of interior muscles that I'm not going to be asking about. There are some muscles, for example, such as the rhomboids, pterygoids, that I may say, hey, where are they located as far as body part? For example, another one would be lumbricals or the dorsal interosseous muscles, but I'm not going to actually have them labeled on the practical exam. Now, if you look on the tutorial that is online, then it's got an interactive exercise that might help you practice this, make it a little bit easier. For the face-to-face people, okay, you're going to have the models to play with, so that's going to help. For my totally online people, you really need to find some actual photographs of some models from different angles if you can't see a 3D model, and work from there. There are some models online that are fairly decent. The problem is so many models correlate poorly to some of the photographs and to some of the diagrams. A lot of times they just look a little bit different, and they make it really hard to identify some of the muscles. So if you're going to go into some form of a medical field that really gets into muscle naming as far as muscle therapy, occupational therapy, that kind of thing, you may want to invest in one of the models to study a little bit deeper than what we've gone. I will see you in chapter 12.